Thank you to Brilliant.org for supporting PBS. What makes us human? Is it the way that we walk, or talk, or think, or invent? Is it the cultures that we create and transmit? Is it the way we interact with our environment? Is it all of the above, or something else entirely? We've been asking this question for a long time, from a lot of different perspectives, from anthropology to religion to psychology to philosophy, rarely finding simple, clear-cut answers. But over the last few decades, an entirely new method of introspection has emerged. We've become able to search our DNA for the hallmarks of human uniqueness at the genetic level. So you might have heard that our genes are around 99% identical to those of chimpanzees, our closest living non-human relatives. So what's in that remaining 1% that encodes humanity? As simple as the question sounds, answering it has proven to be a decades-long quest that is still far from over. And along the way, we've had to change how we think about genomes altogether. Because in the search for the genes that make us human, some of the most important answers were hiding not in the genes themselves, but in what was once considered genomic junk. Now, genetics can sometimes feel like a part of modern science that's easy to take for granted. But compared to paleontology or geology or anthropology, genetics is still a very young field. I mean, even the now iconic double helix structure of DNA was only revealed to the world in 1953, which is within living memory for some people. And the search for human genetic uniqueness only really began in earnest in the 1970s, when scientists became able to zero in on individual DNA sequences. At first, we assumed that the big important differences between us and chimps, the sequences responsible for humanity, would be mostly or entirely found within genes. Technology at the time meant that deciphering even short stretches of the A's, T's, C's, and G's, the bases that make up our DNA, was time-consuming, tedious, and expensive. Human genomes are over 3 billion base pairs in size, so the idea of sequencing and studying the whole thing seemed like a futuristic sci-fi dream for much of the second half of the 20th century. So instead, we had to carefully pick individual bits of the genome to analyze. And because of that, it seemed natural to start with the ones that actually code for proteins, aka the genes. Now, there was also a pervasive idea at the time that genes were where everything that was biologically important happened, and the rest of the genome could be largely written off as non-coding, non-functional junk DNA. And this approach of looking at the genes did reveal some key differences. Like in FOXP2, for example, a gene that seems to play a role in the ability to speak. FOXP2 has picked up some important mutations in our lineage since our last common ancestor with chimps, around 6 million years ago. And a number of other genes that we share as our closest primate relatives have undergone changes too, like being turned off in our genomes or being duplicated or changing location. But as cool as those examples are, they alone can't neatly define or explain our humanity. Even early on, researchers noticed that human genes and proteins generally had very few differences from the corresponding ones in chimps. Far fewer than they had expected, and it seemed like far fewer than could account for all of the differences that we see between us. The plot thickened in the early 2000s, when we finally got a good look at an entire human genome. Before the Human Genome Project was completed, some researchers predicted it would reveal around 100,000 genes, a large enough number to account for all of this magnificent complexity. But it turned out that there were only around 20,000 to 30,000 genes, and these genes made up only 1 to 2% of the genome. It was non-protein coding DNA, the so-called junk, that represented most of the rest. Okay, and here's what bothers me. We have fewer genes than grapes or water fleas. Like, I don't even know what a water flea is, but grapes are the things that like my kids would throw on the floor from the high chair. They have more genes than I do? That's how it's gonna be? Okay. My point is, this is both humbling and confusing. I mean, how could we humans be so complex if we have so few genes? And they're mostly the same as those of our closest living non-human relatives. Well, a few years later, in 2005, the Chimpanzee Genome Project also produced its first draft sequence. Having both human and chimp genomes, as well as other more distant relatives, like mice, allowed us to compare not just our genes, but our non-coding regions too. See, some experts suspected that there were sequences hidden in the junk DNA that were actually functional. Not because they directly coded for proteins like genes did, but because they somehow regulated the activity of those genes. And changes in those regulatory sequences that were unique to humans could be just as important, or even more so, than changes in the genes themselves. After all, genes are basically just biological tools, and you could use the same set of tools to build a table, a chair, or a figurine. It all comes down to how you use them. To sift through all that non-coding DNA for sequences with actual functions and separate them from the junk, we looked for signs of conservation across vertebrates. See, all DNA sequences pick up random mutations, And if a sequence is doing something important, that's generally bad news because mutations often disrupt that function. So over evolutionary time, natural selection works to purge those mutations, keeping the important sequences very similar, even between distantly related species. Much more similar than junk sequences, which can accumulate random mutations and diverge between species more quickly because they have no functions to maintain. And in 2006, researchers identified around 5% of the human genome that was non-coding but tightly conserved across species, which means some of it probably plays important regulatory roles. Think of these sequences as switches that control the activity of genes. And those switches have been around in the non-coding DNA of vertebrates for a long time, and are generally very similar between species. But in humans, thousands of them have been tweaked, altering the way they work and how they control our genes. Scientists call them human accelerated regions, or HARs. Many of them are associated with brain and nervous system genes, changing how those features develop compared to our closest non-human relatives, like chimps. Okay, so search over, right? Humanity comes down to some mutations in the 1% to 2% of the genome that's made up of genes, plus a bunch of tweaks in the 5% that regulates genes. Job well done, everyone. We can go home. Well, not quite. In a paper published in 2022, researchers announced that they had found yet another previously unknown layer of the genomic onion. They expanded their search to the rest of the genome, the more than 90% that is both non-coding and not conserved. And in that bubbling pool of fast-mutating junk, they found something incredible. Entirely new regulatory sequences that are unique to us. They'd been forged from sequences that used to be junk DNA. They weren't conserved in other species, so we'd missed them in our previous searches. The researchers call them human ancestor quickly evolved regions, or hackers. Good work, marketing team. Hackers were about as unique to humans as it's possible to get. In other species, many of those sequences don't seem to do anything. But in our ancestors' genomes, they'd been converted from junk to functional. And what's more, these hackers were the fastest evolved regions of the entire human genome. They had emerged quickly sometime after our lineage split from the chimp lineage, but before we split from our close relatives, the Neanderthals and Denisovans. Okay, but what do these hackers do? Well, the nearly 1,600 hacker sequences are most commonly associated with regulating genes involved in the immune system, the gut, and the brain. Now, the immune system evolves rapidly because it's locked in a never-ending game of cat and mouse with pathogens that it has to constantly adapt to. But the gut and brain connection pointed toward a long-standing idea in anthropology, the expensive tissue hypothesis. This hypothesis suggests that the reason our brains are unusually large and our guts are unusually small, compared to other great apes, is that both of these tissues require a lot of energy. I would not call my gut small, but that's what we are. It was an evolutionary trade-off, and our ancestors went in the direction of big brains, shrinking our guts to balance out the energy cost. So finding that many hackers affect the expression of both brain and gut-related genes potentially makes sense when we look at our differences with chimps. We have come a long way, from our early assumptions about the importance of genes to our more recent realization that non-coding DNA might not be totally useless after all. Maybe the thing that we've really found in our search for the genes that make us human is that it's actually the genetic raw material, the junk DNA, that's most useful to have in your genomic back pocket. It's like the stuff in your junk drawer at home that you keep around, like not because you need it right this second, but because one day it might come in handy. Suddenly, it's no longer junk. Ultimately, the search for human uniqueness at the genetic level has turned out to be more difficult than we'd hoped, more complex than we'd imagined, and more humbling than we ever expected. Because even though the differences between us and chimps might only be a tiny fraction of our DNA, our complexity may not actually be about how much unique DNA we have, but instead about the still murky ways that DNA operates. And in that sense, the search for what makes us human still has a very long way to go. Thank you to Brilliant.org for supporting PBS. Brilliant is an online learning platform for STEM with hands-on interactive lessons. Brilliant is for curious learners, both young and old, professional and inexperienced. Brilliant courses teach you how to think via interactive lessons and problem-solving activities and exercises, and solve problems with interactive lessons in STEM. Today's world is unpredictable, but by learning something new every day, you gain the knowledge to interpret information around you. For example, Brilliant.org offers a course called The Knowledge and Uncertainty. This course gives you tools for managing uncertainty and interpreting information. You will learn cutting-edge mathematics like information theory, Bayesian networks, and causal inference, but without calculations getting in the way. The emphasis is on applying these ideas to deal with the uncertainty in your life. To learn more about Brilliant, go to brilliant.org/eons. Riley J. Mangan, a Ph.D. candidate at Duke University, helped us with this episode. And our staff writer, Farhan Mitha, had a chance to record an interview with him about his work on human evolutionary genomics. And we're making that fascinating discussion available for our patrons. Check it out now over at patreon.com/eons. If this got you thinking about how we became us, be sure to check out our human evolution playlist that covers topics like our place in the primate family tree and when we lost our fur. Also thanks to this month's genius eontologists.
Gail Brown, Juan M., Jaxie Weiss, Melanie Lamb Carnevale, Raphael Hassa, Annie and Eric Higgins, John Davison-Ing, Jake Hart, and Colton. By becoming an Eonite at patreon.com/eons, you can get fun perks like submitting a joke for me to read. Here's one from Stephen Rooney. What's 10 meters long, has scutes on its back, and it hurts like heck when you step on it? A Legosaurus. That's a good one. It's like, if there's a bad Lego dinosaur joke, I have not heard it. And as always, thank you for joining me in the Adam Lowe studio. Subscribe at youtube.com/eons for more adventures in deep time. The Expensive Tissue Hypothesis. It sounds like the name for a prog rock cover band. I would see that band. The Expensive Tissue Hypothesis. They're really good. You got your Yes, you got your Pink Floyd, Emerson, Lake and Palmer, they're all in there. LOL.